Welcome to the AtlantECO podcast, where we talk about the science behind the AtlantECO project, the Atlantic Ocean, and the human adventures experienced along the way. Here, we have conversations with guests from around the world who work together so that we can better understand, manage, and protect the ocean. So let's embark on the journey of AtlantECO and discover the world that lies above and beneath the surface of the beautiful Atlantic Ocean. Welcome back to the AtlantECO podcast. Today we discover another region of the Atlantic with our guest, Paula Huber. She was on board of Tara during the very first AtlantECO-centered studies of the Mission Microbiomes, which is the latest expedition of the Tara Ocean Foundation. And she will tell us about her experience and explain what scientific questions the team has been tackling while at sea and also up the Amazon River. So Paula, welcome to the podcast. I am so happy to have this conversation with you today.
Hi, Eloise. Hi, everyone. It's a great pleasure to be here today and share with you this amazing experience.
Brilliant. So to begin each episode, and before we get into the depth of today's subject, we do like to get to know our guests, to understand how their connection to the ocean started and what their journey has been to where they are today. So, Paula, can you tell us about you, your story, and what you do today?
Well, Alice, I must to say that my connection with the ocean is really recent, at least in the academic. I born and brought in Argentina, in a city near the Paraná River, which is a really big river. And all my childhood and adolescence were in close connection with the river. Then I moved to another city in Argentina to study biology. And I remember the first time that I saw a drop of water in the microscopy, and I became fascinated when I saw the phytoplankton. This drop of water came from a freshwater lake, and I am pretty sure that it was in that moment where my interest in the aquatic microorganisms started. So I did my PhD studying the most tiny and abundant autotrophic organisms, which we call picocyanobacteria. The picocyanobacteria are photosynthetic bacteria that live in almost all the aquatic system, from the ocean to river to the lakes. But I was particularly focused in the community from freshwater system. Then I back to the city where I'm born with a postdoctoral position to study the bacterial community in the Paraná River. So even though those years I was mainly focused in freshwater system and not in the ocean, microbes from both environments share many ecological features. And I apply several technical tools and theoretical approach that are used in marine science to understand the life of these organisms in the freshwater system. After that, the last year, I moved to the Universidade Federal de São Carlos in Brazil to work as a postdoc in the framework of the AtlantECO project. And in August of this year, I was invited to participate in the first leg of the Tara Mission Microbiome. So I know about Tara and its mission before. So you can imagine what exciting I was, but at the same time, I was really, really nervous about this possibility. This was going to be the first time that I would move from the river to the ocean. During 2 months and a half, we sailed through the tropical Atlantic Ocean to the Amazon River. And it was one of the most amazing experiences in my life. I love the ocean, and I love the life in the ocean as much as that. After this, I participated in another leg from Ushuaia to Buenos Aires, And then at the beginning of this year, I crossed the Atlantic from Chile to South Africa on board Tara.
You were extremely lucky to be on board of Tara for so many of the legs. So we have other episodes that cover the other legs. So we've heard from some of your colleagues, or we'll hear from some of your colleagues in other episodes of the podcast. Today, we are here to discover about your first experience during one of the scientific studies from the very beginning of Mission Microbiomes in the Atlantic Ocean. It took place more than a year ago, as you said, and you were on board for the first 3 legs of the AtlantECO scientific studies, having boarded in Martinique and then gone to Macapa, onto Belém, and then finally Salvador de Bahia. So, to start with, could you tell us what scientific questions you were looking to address during all of these legs, why it is important, and how it will contribute to increasing our knowledge of the Atlantic Ocean?
It was a really ambitious expedition, and it was mainly dedicated to study the microbiome, the oceanic current, and the pollution in the Western tropical Atlantic Ocean, which is influenced by the Amazon River. As you may know, the Amazon River is the largest river in the world. And this river is particularly important because it discharges a huge amount of fresh water into the Atlantic Ocean. It was estimated that the river discharges about 200 million liters of water per second in the ocean. As a consequence, this water inflow forms a very dynamic surface plume, which is a surface layer with low salinity that is spread for thousands of kilometers from the river mouth. All this fresh water coming from the river not only affect the salinity and the temperature of the Atlantic, but also it transports sediment, nutrients, and material from the Amazon forest. So this variation in the water condition, of course, has a strong influence over the microorganisms that live in the ocean, and also over the biogeochemical cycles at a global scale. So one of the objectives of this expedition was to increase our knowledge about how the Amazon River affects the Atlantic Ocean microbiome. Before this mission, there were previous expeditions that explored this water. From these studies, we know, for example, that the phytoplankton and bacterial community from the plume are different of those from the open ocean. However, this study were mainly focused only on the surface water. So on board Tara, we took samples from the surface, but also from different depths. So now we are able to discover what is happening in the microbial life below this freshwater layer. As I mentioned before, another important goal in this expedition was to study the impact that the river has on the ocean plastic pollution. The Amazon River moves through big Brazilian cities, carry a lot of plastic to the ocean, and most of the plastic that is present in the river fragment into small pieces that we call microplastic. And the microplastic are then transported by the ocean currents, and some of them pass through regions of high biodiversity where they can impact the marine life. During this expedition, we quantified the amount of plastic in the river, in the plume, and in the ocean. We also sampled the microbes that live on them, which we call plastifer.
Excellent. And can you tell us maybe a little bit more on how this is studied? So what did you have to do? And what will happen next with those samples and the data collected?
Yes, during 2 months and a half, we navigate the Atlantic Ocean, the Amazon plume, and the river on Tara. To study the microbiome, we deploy different instruments and systems to collect water at different depths, which was processed on board. You can imagine what different those environments are. It was a big challenge because in each environment we should adapt different sampling strategy. For example, in the open ocean, we sample deep water, reaching 1,000-meter depth, but in the plume, the maximum depth were not more than 40 or 50 meters. In fact, the sampling in the plume was particularly challenging because this zone of the Atlantic Ocean has the influence of different oceanic currents which are acting all together. And as a consequence, the plume changed in its shape and size very fast. So to define the sampling site, we should use different strategies. For example, every day we receive from the scientist that was online satellite images showing the surface salinity, and this helps us a lot to define the area where we should go. And then in the morning of the sampling day, we use the information from different instruments on board Tara to know the in situ salinity and depth and found the correct location to sample. The river was a completely different scenario. The water are brown because of the high concentration of particles, some of which had this similar size of microorganisms. So it's difficult a lot, the sampling, particularly when we use the plankton nets and also when we filter the water. We pass to collect and filter hundreds of liters of water in the ocean to 10 or 20 liters in the river. Also, besides all the samples that we took to study the microbiome, during this leg we measured a large number of environmental parameters to study the chemical variables and deploy different instruments to characterize the physical property of the water. To quantify the microplastic and study the plastifer, we use a specific nets. We take samples from the surface. We deploy these nets during 1 hour, in general during the land, and then we collect, we characterize, and storage the plastic. During these 2 months, we took a huge amount of samples, which will allow us to study the surface and the deep microbiome using different techniques, such as molecular technique and microscopy techniques, and also to study the ocean plastic pollution. All these samples were processed and almacenated on board, and when we arrived to Salvador, those samples were sent to different laboratories that collaborate in the mission, which will be analyzed in the next month. So I like to describe this part of the expedition as a really dynamic and innovative sampling campaign. Each sampling station was particular in its challenge, but also in its wonderfulness. We should adapt to this dynamic and be part of the system in order to better understand them.
Excellent. It sounds like a lot of thinking whilst you're doing everything. You have to adapt and be flexible, and a lot of samples collected and now being processed in the different labs in the AtlantECO project. So each of the legs of the expedition is dedicated to a woman that is or was involved in marine sciences. And in this case, the legs were dedicated to Sigrid Neumann-Leitao. Can you tell us a little bit about her and her contribution to the field of marine sciences?
Sigrid is a Brazilian researcher and professor at the Universidade Federal de Pernambuco in Brazil. She was a pioneer in plankton research in the north of Brazil. During her research career, she was mainly focused in the study of zooplankton. She contributes a lot with the knowledge about the tropical aquatic ecology in Brazil. In fact, she was one of the first women in participate in Brazilian oceanographic expedition, which was dedicated to explore the tropical Atlantic plankton. I think that dedicated part of the mission to Sigrid is an important way to recognize her work and contribution in the knowledge of the Atlantic plankton community and in science in general in Brazil. It was really nice because before this expedition, we organized two workshops where we invite different researchers which had worked in the Amazon plume to share their experience. And Sigrid participated in the second workshop. We announced in vivo that this will be dedicated, that this mission will be dedicated to her. All her students were in the meeting and she was very excited and really grateful for the news.
That's amazing and great to be able to actually tell the person that her work is being recognized in such a way. Maybe finally, we can talk more about your personal experience on board of Tara. So could you share with us what it was like for you, what your life on board was like, the memories you've brought back from that region of the Atlantic, and also, of course, the memories you've created with all the crew and the team on board?
Yes, well, as I told you before, this was the first time in an oceanographic expedition, and it was, in fact, one of the most amazing experiences in my life. We were 40 persons on board from different parts of the world, living together in the ocean in a 36 per 10 meter boat during 2 months. So yes, I remember that when I arrived, I didn't know any person on board from before, but after a few weeks, we were like a family. We shared a lot of good moments and conversation. Every day we were plenty of activity, of course, scientific activity, but also another kind of activities that clean the boat or prepare the lunch or the dinner service. Yes, and the life on board was really, really fantastic. One of the moments that I enjoyed more was the sunset at 6 p.m. more or less. Everyone stopped what they were doing, and we found all together outside the boat to see the sun entering the ocean. I remember that the station days were a bit hard because we start very early in the morning at 5 a.m. and we finish at night. And of course, someday you felt a bit tired. But an amazing thing to sample in a sailing boat is that you are in close connection with the environment. So in several stations, while we were sampling, some big animals and whales and dolphins arrived to Tara to see what we were doing, and it charged us full of good energy to continue working. Another good memory that I have is one day that I wake up early in the morning. I took my cup of coffee and I went outside, and the water was completely changed its colors. It was brown, and we were sailing the Amazon River waters. So everybody knows that the river is really big. I know before that the river is really big, but it's only when you are there that you really realize the size of the river. So yes, and this day a pink dolphin welcomed us.
That sounds amazing, and I bet you also created lots more memories on your further adventures on board of Tara, further down the coast, and then when you crossed the Atlantic So it must have been also some amazing experiences with beautiful sights. That's our conversation for today. So Paula, I have to thank you for taking the time to talk to us and to share the experience that you had on board of Tara. It was a real pleasure to talk to you.
Thank you so much.
As I mentioned, we are recording with all the colleagues that were on board in the different legs of the Mission Microbiomes, so make sure to come back and check out for future experiences shared on the podcast. We hope that you've enjoyed today's episode and look forward to seeing you next time. You can follow the AtlantECO project on our website on www.atlanteco.eu, and you can also find us on Twitter, Instagram and Facebook. All the links and information on the project and on today's episode is in the show notes. AtlantECO is a project funded under Horizon 2020, a European Union research and innovation programme.